Hi everyone, this is Miss Nguyen, and today we're going to cover gene expression, which is the process of converting the information that's in your DNA into ultimately the proteins that help make up your body's tissues, as well as the traits that you observe in the body. This lesson is going to review chapter 9.1 and 9.2, and in this process we're going to review these key concepts. Now, what do genes code for, and how does DNA code for cells and bodies? How are the cells and bodies made from the instructions in DNA? Well, basically, DNA encodes the exact sequence of amino acids that you put together that will lead to a specific protein. The specific amino acid is going to affect the shape of the protein and thus what the protein will do, whether or not it forms structures in your cells or whether or not it forms enzymes that do work in your cells. Ultimately, those proteins affect how the cells are formed, as well as what they end up doing. And those are what traits you end up observing in someone's body. So if someone has an inability, for example, to process milk, that's because they don't have enough of the enzyme that breaks down lactose sugars in milk. If someone is diabetic, that's because they have an inability to process the protein insulin, or they have an ability to create the protein insulin that helps them handle sugar that's incoming from their food and moving into their bloodstream. Or if you have black hair rather than blonde hair, that's because you produce a melanin protein much more strongly in your hair follicles, your hair cells, than someone who has blonde hair. And all of this protein production is coded from the gene recipe in our DNA. So the central dogma is essentially the general flow of how proteins are made based off of genetic information. So DNA can either be replicated to form more DNA, which you'll usually use if you're trying to form a new cell, or DNA generally, when your cell is doing its job, is utilized to make proteins to maintain the cell or to do work. In this case, what we're going to need is we're going to need DNA to turn into RNA through a process called transcription. Then we're going to take that RNA and turn it into protein through a process called translation. And those proteins is what leads to the traits we observe. So, generally speaking, in this case, RNA is the driving molecule that's really essential for mediating the DNA and the protein. And one of the reasons why it's so critical is your DNA is really essential for your cell. It's very critical, it's very important, and you only have the one copy in the cell. You don't want to damage it. And you don't want to move it constantly to a ribosome in your cytoplasm, because that's a lot of work because the DNA is very big. But also when you move that DNA to the cytoplasm, you're putting it at risk for damage. Imagining taking your school textbooks to the beach to study, for example, or by a poolside to study, because I know some students who have done that while they were like on vacations or doing a sport, and then they get water in their textbook and it gets damaged and like, ah, crap, I owe the school money. Same idea of putting your DNA into the cytoplasm every time you just want to make a protein. So instead, what we're going to do is we're going to make some little trash copies called mRNA, which are just going to copy only the recipes you need from your DNA. It's equivalent of if you wanted to do homework from your textbook, but you don't want to damage your textbook or bring all of it with you, you're just going to take a picture of the pages of your textbook that you need to do your homework later. That's the same idea. So I'm going to take my trash RNA copies and use that, make that copy based on the DNA, and then bring that trash copy into the cytoplasm so that it can be read by the ribosome in the cytoplasm to be turned into protein through translation. So that's the idea. RNA is the bridge between protein and protein synthesis, and because of that, we need to know how RNA is made from DNA in the first place. So RNA structure, just a reminder, when you compare it to DNA, is that besides the fact that its function is different in that instead of just carrying information, it's used actively for the production of proteins based off of DNA information. Within the structure of RNA itself, there's some critical differences that allow the cells to distinguish between DNA and RNA. One of them is that it uses a ribose sugar instead of a deoxyribose. That's very important because the polymerases that build RNA and DNA are different, and you're going to make sure they can distinguish themselves and only attach to the right nucleic acids by recognizing whether it's reading a deoxyribose sugar or a ribose sugar. In addition, the nitrogenous bases that are utilized in both of them are slightly different. In this case, RNA uses uracil instead of thymine. So in this case, A goes to U and C goes to G. Okay, so anytime you intend to write a T in RNA, instead write a U in the RNA sequence. And in addition, generally speaking, at least for the processes we're going to cover, and most RNA that you find in a eukaryotic cell, most of that RNA is single-stranded. There are some very small exceptions to unique processes, but when it comes to the stuff we're going to study in this class, in this unit, generally speaking, single-stranded. And there's lots of kinds of RNAs. We're going to only cover 3 of them in our class, but there's actually a lot more different various types of RNA, and we're discovering more all the time. For example, CRISPR is based off of RNA that we didn't realize was in the cell and super important until like 10, 20 years ago. Or we probably knew about it before then, but we didn't realize how useful and important it was until about 10 to 20 years ago. All right, and this is more of a more, I guess, complicated or more complex picture of RNA versus DNA, and it helps you maybe observe that the nucleotide bases, the nitrogenous bases, kind of hang off the RNA sugar phosphate backbone, while in DNA, the nitrogenous bases are paired up with the opposing strand of DNA, the anti-parallel strands of DNA. All right, so there are 3 main types of RNA involved in protein synthesis, but today mainly we're going to go over messenger RNA, which is the key trash copy of RNA that passes on and contains the protein building message from the genes themselves and specifies the order of amino acid sequence, and that it's going to carry that information from genes from the nucleus to the cytoplasm. Later on, we'll talk more about tRNA and rRNA. tRNA is going to assist with bringing in amino acids when we build our protein. rRNA is actually the foundational molecule that helps form ribosomes. But again, these are more critical in a process we'll cover in a later lecture. All right, so getting from DNA to protein requires 2 steps. First is making that trash copy, that copy of the gene, but in an RNA form that we can then send out to the cytoplasm to be read. And then the second step is translation, when we take that recipe mRNA that's temporary and then utilize it to actually build the resulting protein. Now, in bacteria, because they have no nucleus and because they're much smaller and tend to be a little bit more evolutionarily simple compared to eukaryotes, generally speaking, the transcription and translation process are both done in the cytoplasm, and that's that. And the processes I described for transcription and translation is all that happens. However, in eukaryotes, there's a little bit more of an extra step. So one of the things is that the DNA is kept in the nucleus because we want our DNA to be safe. In addition to that, there's an additional process in all of this called RNA processing, and I'll sometimes refer to this in the lesson as RNA modification. And RNA modification or RNA processing in this case does a little bit extra to that RNA copy before it's finalized into the mRNA and then can be utilized to make proteins. And there's some advantages to that additional processing stage that is granted to eukaryotes that do it. So it's a little bit extra work, but also it's going to give us the advantage of, I'll just preview a little bit, it's going to give us the advantage of being able to potentially make more than one protein from a single gene based off of the way this RNA gets processed. All right, so transcription is a synthesis of RNA using information from DNA. And the big one here is RNA is built 5' to 3'. All right, so transcription, and just like DNA, always built in the 5' to 3 direction. But because we're only building one strand of RNA, I only need to build my one leading, it's not a leading strand because there's not 2 strands of RNA, but it's like that one leading strand of DNA being made in one piece. The one piece of mRNA I need to make, I can just build off in the 5' to 3' direction. So what I'm going to use is I'm going to open up and unzip my DNA, and I'm going to open up to the template strand. So I'm going to use the template parent DNA strand in the 3' to 5' direction. It's a little hard to see, but if you track it, this DNA that's being read for the RNA is in the 3' to 5' direction. And that means that the complementary RNA that's being formed is into the 5' to 3 direction. So I just can make that one smooth mRNA sequence from the DNA. Generally speaking, transcription is only about reading and copying the mRNA or an RNA piece that's directly from the gene. So you're not going to copy all of the DNA in one big piece. You're just going to make one short, relatively short RNA piece from just the gene sequence. RNA polymerase for the transcription process, again, we don't need a DNA to fully unzip, we just need the one gene. And because we're only making one strand in the 5'3" easy direction, that means I actually only need one protein for this whole process. I need the RNA polymerase protein, which is an enzyme that's used for RNA synthesis, and it does everything. Unlike the many proteins we need for the complicated process of copying all your DNA, in this case, we just need RNA polymerase to do all the things. It will unzip the DNA and then read the DNA sequence that we need, the one gene sequence that we need, and then form the RNA sequence and join the RNA nucleotides in the 5'3" direction by adding, we're always building the 5'3" direction, meaning we're adding to the 3' end. And it's RNA, so I don't need a primer to start it because it's RNA. The primer for DNA creation is RNA, and I'm making RNA here, so I don't need a primer. I just make my one piece of RNA. And that's that. I'm done. My RNA polymerase, all it does, unzip, build my RNA in the correct direction, release it, I'm done. So that's the idea of transcription. We just unzip a little bubble, a transcription bubble of my DNA in just the one gene that I want to use. And I'm just going to pop off using the RNA polymerase a copy of the RNA in the 5'3" direction off the one template DNA strand that I want. There's different DNA polymerases for each type of RNA, but mainly today we're going to talk about the formation of mRNA, but all the RNA is formed in the same similar process of transcription. However, I did want to also point out that this process of making the RNA as well as modifying and any weird changes we make to RNA, they're generally done in the nucleolus, which is a protected region inside the nucleus in eukaryotes. Okay? All right. So just reminding us, our RNA is based off the DNA sequence, and we're literally building a complementary sequence based off the DNA. And it's just like when we do DNA replication, except when we're doing our complementary strand on the RNA sequence, if we want to put in a T, such as here, we're going to put to match the A that's on the DNA sequence. Instead, we put in a U in the RNA strand that we're building. All right, there are 3 stages to transcription: initiation, which is probably the most complicated thing I'm going to cover today, elongation, and termination. So we're going to initiate, start it, actually build it, elongate, and then terminate, which is end and release the RNA that we formed. So the actual process of initiation really heavily involves what's known as a promoter region. So if you recall, in DNA, not all of your DNA includes the genes, but then there are segments with the gene itself. But what you may not know is this is a gene, and oftentimes the region in front of the gene here, and maybe sometimes includes the gene itself, this region here is a promoter region, the region upstream or in front of the DNA gene sequence that actually helps direct where our RNA polymerase attaches and when our RNA polymerase attaches. Basically, it indicates the starting point for transcription of a gene, and it actually helps control and regulate when you use that gene and when you help make RNA from that gene so that you can make proteins from that gene. All cells have promoter regions in front of their genes, bacteria, eukaryotes, though bacteria in addition have a terminator sequence at the end of their gene, while that doesn't necessarily exist in eukaryotes. So for in the promoter region, the way it works is transcription factors mediate the binding of RNA polymerase and the initiation of transcription. All that means is there are proteins and chemicals and hormones, and they have to basically act as an on switch. And when they bind to the promoter region in front of the gene, so the blue region is the gene and the green region is the promoter, when they bind to the region in front of the gene, they help provide a clicking on point for the RNA polymerase to attach and start reading the gene and forming the RNA transcript from it. Oftentimes, a big part of this promoter region that is necessary for allowing the attachment of the RNA polymerase is called a TATA or a Tata box sequence, which is a great area for allowing the DNA to open, because if you recall, T's and A's have 2 hydrogen bonds, while C's and G's have 3 hydrogen bonds. So when you have a long sequence of just A's and T's, it's actually relatively weaker in terms of hydrogen bonds compared to a region that has mixed C's and G's in there, or only C's and G's. So it's a lot easier to start unzipping the gene and reading the gene to make the RNA. Okay, so that initial complex where you've unzipped the gene and you helped with your transcription factors bind the RNA polymerase so it starts reading the DNA and turning it to RNA. This whole structure is the transcription initiation complex. All right, so our transcription factors help bind to the DNA and turn it on, and then our RNA polymerase starts reading it and forming the RNA. The transcription factors are things that help trigger the gene to be used when it's appropriate. For example, when you hit puberty, a bunch of hormones are flowing all throughout your body, and those hormones will then trigger tissues in your body start maybe producing and turning on the genes that help produce extra hair or extra muscle proteins or extra growth proteins that help build your tissues to get you taller and things like that. So why do promoters matter? They control where your genes express, and the proteins you make define your cell. So, for example, you don't want the proteins that calcify bones to be expressed in your brain. One of the things I did in college was I actually studied promoter sequences in plants, and you could actually track promoter sequences by randomly grabbing DNA pieces, sticking it in front of a gene that turns cell tissues blue, and then we would test different promoter sequences to see where they expressed things inside a plant, and then we would help figure out if this gene is expressed in the stem of a plant, if it's expressed in the leaves in the plant, or if it is expressed in the fruit of the plant. And by doing this, you can then hack the plant by maybe forcing a gene that helps produce more sugar in the plant to be produced inside the fruit only. So that's one way you might be able to use these promoters to genetically manipulate stuff, for instance. All right, the last 2 parts of RNA production are elongation and termination, and these are pretty quick. So elongation is just that process I described earlier when we've already unzipped our protein and our RNA is just being formed from our RNA polymerase, which as it's moving down the strand, it's unzipping its little bubble of DNA and making my RNA off the 3' end. And when this RNA polymerase moves down to this part of the DNA, it will also open that part of the RNA and then make the remaining part of the RNA transcript. And then finally, we hit termination. So as my RNA polymerase, as it was reading my DNA and moving down the strand, when it hits the end of the strand, it then releases and drops off of this strand and then also releases the RNA transcript. This is termination, the end of transcription. In bacteria, this is signaled by the RNA polymerase hitting a termination sequence, also known as the terminator, in the RNA to release the mRNA. And for prokaryotes, they can immediately use the mRNA that they formed. In eukaryotes, it's a little bit more complicated. So once the RNA polymerase hits the end of the sequence, it's usually just a bunch of like trash A's at the end. And eventually the RNA polymerase is like, it sort of signals to the RNA polymerase to let go. And at that point, the RNA polymerase drops off and then the transcript is then released. But we're going to need to take this RNA transcript and process it and modify it a little bit more before it's actually usable by the eukaryote. Okay? So transcription actually looks like this in real cells, and it kind of forms this Christmas tree-like structure, because what you may not know is more than one RNA polymerase can be working on a single gene at any given time. So this is one gene, and this is the DNA strand. That's the single DNA strand. And all those little bits that you're seeing, all those little branches, each of those is a single RNA being made by a separate RNA polymerase. So this one gene is being made hundreds of times at once by many different RNA polymerases. Each of these separate Christmas trees that you see is a gene that's being read many, many, many times by many, many hundreds of RNA polymerase, all making RNA at once. So when you make your genes available for production, that gene is going to be transcribed very quickly and many hundreds of times so that you can immediately get that protein to be made by many RNA in your cell as soon as possible. And just a reminder, in eukaryote cells, that transcription process occurs in the nucleus, and in prokaryotes, it occurs in the cytoplasm, and for eukaryotes, it's going to need a little bit of an extra step. So the genetic code from our DNA, which consists of only 4 bases, is going to lead ultimately into 20 amino acids, into encoding 20 amino acids in our proteins. So how many nucleotides correspond to an amino acid? We'll go into the math of why this is in the future, and we'll go into why that's significant in the future, but generally speaking, all you really need to know for now is that in terms of the flow of information from our DNA to our protein is that there is a 3-letter codon sequence in our DNA and thus in our RNA, and every 3 letters specifically relates to and corresponds to an amino acid that's eventually going to be in the final protein. And the order of your 3-letter triplet or codon sequences that are found in your ultimate RNA that you form is what's going to ultimately lead to the specific order of amino acids found in the final protein. Now, I mentioned in eukaryotes, there is an additional process once the transcription is made before you can actually successfully make final mRNA that can be sent out for translation and conversion into protein. And this process is called RNA modification, also sometimes known as RNA processing instead of RNA modification. Now, why do we do this? Well, there's actually a really big advantage to it, and we're going to go over that. So in this case, in eukaryotes, the actual RNA that is made from pre-mRNA is it's not fully usable RNA because the DNA sequence itself has different regions of it where some of it involves actually used DNA called exons, and some of it uses kind of what we call junk or unexpressed DNA called introns. So in order to use our RNA, we're going to need to cut out the used RNA, the unused RNA, and then we're going to need to add a little bit of an additional 5' cap and 3' tail to help this RNA do its job and go to the correct places and to protect it. All right, so the first step we're going to need to do is cut out all the, quote, junk DNA. Eukaryotic genes are not continuous, and oftentimes that means there are sequences in the RNA that is formed from the DNA itself. So this is not the RNA, but this is the DNA, and this DNA has sections of it that are the real gene that has the coding DNA that's going to provide the sequence that we want for the protein we're going to make. And there are sections which we call the junk sequence, and it's not always junk, but at least for the mRNA that we're trying to form, it's sequences that aren't useful to the mRNA we want. It's the in-between sequence, not the used sequence. So the exons are expressed and the introns are in between the expressed section. This confuses students. They think introns stay in. No, the introns are the ones that leave and the exons are the ones that stay in the RNA that you're going to finally make. Okay, so what you're going to need to do is cut out all the introns and then join all the exon parts together. All right? And then on top of that, you're going to add some, like I said, 5' and 3' tail, which you can see in this image, some protective 3' and 5' ends. So RNA splicing is the process of cropping out those introns and joining together all those exons. So the RNA, before it's been fully processed and has those introns removed, we call this the pre-mRNA. It's the initial RNA transcript in eukaryotes that still has those non-useful introns. And splicing is a method of which we remove those introns. The whole splicing enzyme sequence involves proteins and more RNA types called spliceosomal RNA, but we're not going to go into it. Just understand that there is a kind of a more complicated process involving your cells, proteins, and RNAs to help remove the introns, but at the end of the day, the introns are going to be removed and the exons are the only part of the RNA that is going to remain to make the useful mRNA. And you want to make sure this mRNA is correct because if we splice it wrong and we leave, let's say, excess introns and stuff that we may not want, this can have an effect by making a reading frame, for example, having this extra G here will throw off and push all my RNA sequence down here, down one, and it will drastically alter the resulting protein that will form. So maintaining and doing it correctly to maintain the reading frame and the correct codons is really critical in the splicing process. And in addition to that, the reason why we like splicing is because it introduces the ability to do something called alternative splicing. So alternative splicing is a special process where we said that in DNA there are exon sections and introns for a given RNA that you may want to make. Well, what that means is from this single RNA transcript, depending on how you splice it with alternative splicing, in some genes, not all genes, but some genes, that splicing can be done in different ways to form different alternative versions of mRNA. Why is that important? Well, for eukaryotes, oftentimes there will be a lot more proteins that exist in a single cell of a eukaryote, or not single cell, in the entire body of a eukaryote than their genes. For humans, there are about 20,000 genes, but there are far more proteins found in our body than 20,000. And the reason why that's allowed is because through alternative splicing, the one gene that is transcribed into a pre-RNA can lead to many different alternative versions of the final mRNA, and thus many alternative proteins from one gene. It lets one gene lead to many proteins. Now, students get confused. What's the difference between splicing and alternative splicing? All genes need splicing to remove the introns. And some of the genes that are in eukaryotes, though, can lead to many alternative versions of the RNA. But there are some genes in eukaryotes where the splicing only occurs and leads to only one version, and there are no alternative versions, no alternative splicings that can be made. There's only one final RNA that can be made. So splicing is the process itself, and alternative is just referencing the fact that some genes have the ability to splice and actually form multiple different RNA versions from one gene. All right, and finally, on this RNA, we're going to add some additional things to it. We're going to add a 5' cap, which involves the modified guanine cap, also known as a GTP cap, to the 5' end, and this guanine cap actually helps the mRNA bind to and hook into the ribosome so that it can be read. And it also prevents the RNA from being broken down in the cytoplasm, because remember, the cytoplasm is kind of dangerous to RNA, and the RNA may break down pretty quickly, so you need a little bit of protection so that RNA can last long enough to reach a ribosome so it can make proteins. And then we're going to also add what's called a poly A tail, that 3' sequence of A's at the end to help allow the exportation of the RNA into the nucleus, and also add a little bit of extra protection onto my RNA sequence so that it can escape. All right, so this is sort of the overall image of the transcriptional modifications, or RNA modification. And in this process, we're going to crop off our introns, leave the exon, and add a 5' cap and a 3' tail at the end. And this is the final RNA that is created after transcription and after RNA processing in the eukaryote. And this is the mRNA that we're going to be able to send out to the cell to help to allow the ribosomes to read and turn it into a protein. All right, so the full process then, at least in eukaryotes, not prokaryotes, is DNA through transcription leads to pre-mRNA, and through RNA modification, through splicing and adding those caps and tails, we're going to get our actual final mRNA, maybe alternative and more than one version of that mRNA, and then we're going to be able to later on translate that into a protein. All right, this is a video if you need more explanation beyond this video, although this video is quite long. This is just kind of a really rough summary of what I covered, and these are the vocabulary terms that we covered in this lesson. All right, that concludes the lesson. Bye.